Okay, yeah. So when Arvid was telling us why Chicago is such a good place to have this meeting and channeling John Maynard Smith and saying how horrible Chicago is, how people say inspiringly stupid things here, I noticed that he didn't mention the reason that Maynard Smith was in Chicago in the first place, which I presume is because it is one of the historic centers of gravity of population and quantitative genetic theory. So in that spirit, and hopefully not the others, I'm going to talk about this. I'm going to talk about a rather abstract quantitative genetic model that Pavitra and I have been thinking about, in which we're finding to have interesting consequences for population and evolutionary genetic processes. So I'm going to consider a biological system with the output of which is quantitative, so some measurable outcome. The system's going to comprise some number of components, and these components, whose quantitative contributions to the system I'm going to denote by y1, y2, and so on, are going to combine additively to determine the overall value of the system Z. So this is a very general model of a biological system. One particularly concrete instantiation of this model from sort of my hometown of complex trait genetics is that we think of the system as just a quantitative trait, and then you think of the components of the system as the contributions to that trait of different regions of the genome, such as the chromosomes. But another possibility is that it's a gene regulatory network, and we think of the components as perhaps modules within this network. It might be an interspecies mutualism, where the components are different species contributions. And I'll talk about an example in that line later. But this model obviously encompasses many, many different biological systems. And I'm going to assume that the quantitative outcome of the system is under some kind of constraint. So it's under stabilizing selection for an optimal value. And I'm thinking of the system and its components, sort of as we now know of many, many traits, as highly polygenic, so affected by mutations at a great many loci throughout the genome. And with such a large mutational target, the persistent influx of variation due to mutation will lead the system's value to be distributed something like this in the population. And since the outcome is under stabilizing selection, the dynamics are very straightforward. The mean value of the system is going to move to its optimum, and it's going to stay there. The selection's maybe going to try to reduce variation in the system, and that variation is going to be replenished by mutation, and so we're going to be held at some kind of balance. If we want to quantify that, we need to specify the parameters of the model. So I'm going to arbitrarily code the optimal system value as 0. I'm going to assume that the variance for the system outcome is Vg. So I'm ignoring environmental variance here. This is just the genetic variance for the system. And I'm going to specify that the fitness function be Gaussian with a variance equal to Vs. So larger values of Vs correspond to weaker stabilizing selection on the system outcome. And under this specification, the outcome of the system is just a quantitative trait under Gaussian stabilizing selection. And so we know that its mean value behaves as an Ornstein-Uhlenbeck process. Okay, written here as a stochastic differential equation. And since I'm going to be working with these a bit in this talk, I'm just going to explain what the terms in this equation mean. So I'm going to jig them a little. And we see that the change in the mean value of the system has a deterministic component due to selection, and it has a random component due to drift. In the deterministic component, there's a selection gradient. So if the overall system's value is above its optimum, then selection is trying to bring it back down to the optimum. If it's below the optimum, selection is trying to push it back up. That selection gradient is stronger if the strength of selection is stronger, and if the overall mean value of the system is further away from its optimum. The response of the system mean value to the selection gradient is proportional to the genetic variance for the system. And the random term due to genetic drift is a Brownian motion. Okay, so Wt here is a standard Brownian motion, and the magnitude of this genetic drift is larger if the effective population size is smaller and if the genetic variance for the system is larger. Okay, so how does this process play out? Well, if we start the system with its overall mean value below the optimum, selection pushes the mean value up to the optimum and subsequently holds it there in a narrow window around the optimum. Okay, so from the sort of system-wide perspective, the dynamics are very, very straightforward. But remember, we're interested here not in necessarily in the mean value of the system itself, but instead in the behavior of the components of the system. So that's what we want to work out. We want to work out dynamical equations that describe the change in the mean contributions of each component to the system. Right, and so with Pavitra, we've found a decomposition of the overall system's Ornstein-Uhlenbeck process into a system of coupled stochastic differential equations, one for each component in the system. And so to explain what's going on here, let me just pull out the equation for component I. Right, so once again, the change in the mean contribution of component I to the system has a deterministic component due to selection and a random component due to drift. In the deterministic component, there's a selection gradient that's common to all of the components. So if the system's overall mean value is above its optimum, every component is under equivalent selection to try to pull the system mean value down towards the optimum. The response of component I to this selection gradient is proportional to its contribution to the system's genetic variance. So Fi here is the fraction of the overall system's genetic variance that is contributed by component I. Each component is subject to an independent drift process, right? So the Wi's here are independent Brownian motions, and component I's drift process is larger in magnitude if, again, the effective population size is smaller, and if component I contributes a greater fraction of the system's genetic variance. Okay, so we have this system of coupled stochastic differential equations, coupled because of their joint dependence on and determination of the system-wide mean value ZT. And the goal is to kind of decouple these equations, to isolate the behavior of each individual component in this process. Okay, so to begin to do that, I'm going to just once again pull out the equation for the ith component, and I'm going to sum together the equations for all of the other components and index the result by minus I. So we now have a system of two coupled stochastic differential equations. Okay, and so to decouple these, I'm going to basically just apply some relatively simple arithmetic operations. So the first thing I notice is that the selection terms of these two equations look structurally similar. They differ only in these factors Fi and 1 minus Fi, the fraction of the system's genetic variance contributed by component I and the fraction contributed by all of the other components. So I'm going to divide the equations by their respective factors, and now the selection terms look the same. So if I subtract the one equation from the other, the selection terms fall out, and I find that the scale difference between component I and the rest of the components is a combination of two independent Brownian motions, and therefore a Brownian motion itself. Okay, I now remember that component I and the rest of the components have to sum to the system-wide mean value, so I can replace the rest of the components in this lower equation with the system-wide mean value minus that of the ith component. And if I tidy things up, I'm left with a stochastic differential equation specifically for the mean genetic value of the ith component of the system. And I see that it is composed of two Gaussian processes. The first is the Ornstein-Uhlenbeck process that governs the convergence of the system-wide mean value to its optimum. And the second is a Brownian motion. These two Gaussian processes turn out to be independent of one another. And so component I's mean value is itself a Gaussian process. And importantly, we know that the Ornstein-Uhlenbeck component of component I's behavior is eventually going to be approximately constant as the trait moves to its optimum and stays there. And so that means that component I's mean value is eventually going to come to behave as a Brownian motion. Okay, that is, it's going to be free to drift far from its initial value in a way that is qualitatively identical to neutral genetic drift, as if the system was not under selection at all. We can see this played out in simulations similar to the ones that Pavitra showed earlier. So here is the system-wide mean value. The system is under stabilizing selection, and so it's held within a narrow window around the optimum. But if I pull out component I and the rest of the components' behavior, I see that they behave in the sort of random drift-like fashion, right? They're free to drift far from their initial values, just in a way that's sort of mutually compensatory, so that they always sum to approximately the system's optimal value. We can measure the rate of this drift process by imagining the process played out many replicate times and measuring the variance of component I's outcome at some point in time across these replicates. And since component I's mean value is behaving as a Brownian motion, this variance is straightforward to compute. It grows linearly in time at a rate that depends on the effective population size, the system's genetic variance, and the fraction of that genetic variance contributed by component I. Okay, so component I is behaving as a Brownian motion, as it would if there were absolutely no selection on the system at all. But how does its rate compare to what it would actually be if there were no selection on the system? Right, well, we know what the rate of drift would be if there were no selection on the system. It would also grow linearly in time. And the difference between the rate of component drift with and without selection on the overall system's value is given by this factor of 1 minus fi. So kind of sensibly, if the component in question accounts for all of the genetic variance of the trait, all of the genetic variance of the system, then selection on the system is going to constrain that component's evolution. Whereas if the component contributes relatively little of the genetic variance of the system, then it would be far more free to drift as if neutral. Okay, just quickly some robustness results that I won't have time to speak about the details of, but this process of system drift we find occurs despite periodic changes in the optimal value of the system, essentially because movement towards the new optimum is absorbed into the Ornstein-Uhlenbeck term in that decomposition, leaving the Brownian motion term unchanged. It's robust to the possibility that the genetic loci that contribute variation to the system also contributes variation to other traits or systems under stabilizing selection. And back mutation between system increasing and system decreasing alleles applies a very, very weak and very, very long-term break on system drift, which is unlikely to be relevant on empirically reasonable timescales. Okay, so that's the sort of general model. And as I said, we're finding that this model has sort of conclusions for very many processes in population and evolutionary genetics. One of them is that it kind of provides a quantitative foundation for and quantification of the theory of developmental system drift. So this is the theory first proposed in developmental genetics that if you have a developmental system with different modules or parts, even though there's constraint on the system value and therefore no divergence between species in the overall outcome, the components of that system can evolve and therefore diverge between species. There are very direct implications for complex trait genetics, right? So the prediction here is that if you have a trait under stabilizing selection, then even though the genome-wide mean genetic value is held constant, different regions of the genome can drift in their contributions to the trait. And so, for example, we've estimated that for human height, which is a trait that has directly been measured to be under stabilizing selection, chromosomes 1 and 2 should differ in their mean genetic values for height by something like 2 meters. Okay, sort of strange conclusions like that, but inevitable under the theory. Yuval-Simons, a colleague at the University of Chicago, and I have shown that this process of sort of chromosome scale drift has important implications for reproductive isolation between species. It generates reproductive isolation even when populations or species are under common selection for the same optimum. Pavitra presented earlier her work showing that this process generates barriers to sex chromosome turnover. Julia Holder, who's here, and Pavitra and I are working, as Pavitra mentioned, on a theory, if we have a quantitative trait under stabilizing selection and some fraction of the genetic variance of that trait is contributed by the mitochondrion, then how do the mitochondria and the nuclear genome co-evolve under that process, and what implications does that have for male and female fitness? But I'll talk now about some strange conclusions that we've reached in the case of mutualisms, and this is work with a graduate student in our biophysics program, Serena Debasai. What I'm going to imagine is a mutualism where I have two species, and each species is capable of producing some kind of public good. So perhaps some chemical that's secreted into their joint environment and which can maybe digest some extracellular resource with the products of that digestion then available to both. So this is just a classic public goods problem. I'm going to imagine that the green species contributes X and the pink species contributes Y, and I'm going to assume a very particular interaction structure where members of these two species interact in pairs. So I'm sort of abstracting away from public goods problems within each population to concentrate on the public goods problem between the two populations. The overall production in a pairwise interaction where the green species produces X and the pink species produces Y is going to be given by Z. Okay, so Z is the amount of the public good available to the two interactants. And there's some benefit to this public good and there's some cost of producing it. Okay, so the benefit depends on the overall amount produced and the cost depends on the amount to each individual in the interacting pair. And the fitness is then the benefit minus the cost. So this is just the general model of a snowdrift game as defined by Duberly et al. In this paper. But there, as is usual in the evolutionary game theory literature, they're thinking of this game being played between pairs of individuals within a population. But here I'm thinking of it being played between pairs of individuals between two populations. Okay, so I'm going to specify for now the simplest possible cost. I'm just going to assume that each additional unit of the public good produced costs each species some common amount S. And I'm going to assume that the benefit takes a sort of stabilizing selection form. So I'm going to assume that there's some kind of agreed upon optimal amount of the public good to produce, right? Too much would be bad, perhaps because too much is toxic, something like that. So there's an optimal amount Z star, and both of the interacting parties agree that that is the optimal amount. So you can think of this as the amount that each would like to be produced if it didn't have to produce it itself and therefore pay the cost. Okay, so under this specification, we have fitness functions of the amount that I produce given the amount that you produce. And from these, we can calculate selection gradients. And assuming that there's some genetic variance within each population for the amount produced, we come to, again, a system of stochastic differential equations governing how the mean value produced within each population changes over time. I can apply exactly the same mechanics as before. But before that, I suppose, if I sum these two equations together, I obtain a stochastic differential equation describing the time evolution of the total amount produced, the total mean amount produced within each species, and I find that it's an Ornstein-Uhlenbeck process that converges to a long-term mean value that's slightly less than the agreed upon optimal amount, and that's because of the costs associated with producing this public good. Okay, but I know therefore that the overall amount produced is going to converge to this equilibrium value, but the interest here is in the sort of division of labor within this mutualism, the amount produced by each party. And so if I take these coupled stochastic differential equations, I can decouple them just as before. And again, I find that the mean amount produced within each species is a Gaussian process composed of an Ornstein-Uhlenbeck and a Brownian process. And that means that because the Brownian components are going to come to dominate the OU components, the mean amounts produced by each species are going to come to drift randomly as Brownian motions. Okay? And that's a particularly strange result here, because remember that there's a cost to producing, there's a fitness cost to producing this good. And so if the mean amounts produced by each species are drifting randomly over time as Brownian motions, what that means is that the mean fitness of each species is also drifting randomly. Okay, so that's a pretty strange result. It's relatively easy to break that result. I mean, it is a result in this particular specification, and it is, I think, a sort of strange and perhaps paradoxical result. But we could, for example, assume that instead of having a linear cost structure where each additional unit of the good costs the same amount to produce, costs the same amount to produce, we might imagine accelerating costs. As you produce more and more of this public good, you have to kind of subordinate more and more important metabolic processes. So you have this kind of accelerating cost structure. And so Serena and I are working through these various cases. In this particular case, you reach an equilibrium where each species produces an intermediate amount of the good. Another possibility is that the two species disagree on the optimum. So one species actually prefers slightly more of the public good to be produced than the other. In that case, the long-term outcome is that the species that prefers more to be produced produces all of the good, and the one that produces less produces none. Similarly, it might be more costly for the one species to produce the good than the other. In that case, even if they agree upon the optimum, it is the species for whom it is cheaper to produce the good that comes to produce all of the good, and the species for whom it's more expensive ends up producing none. So I framed this in terms of two species playing this game, but really all that matters is that the interaction be asymmetric in some sense. So this could be a within-species interaction, as long as the roles within the game are definable and the strategies played by each role are genetically distinct. So for example, you could imagine that this is an example of parent-offspring conflict, right, where the strategy of, say, the mother and the strategy of the fetus are encoded separately within the genome. So here I'm thinking of cases where, of sort of hormone production, like in human pregnancy, the hormone that has been studied from an evolutionary perspective by David Haig, human chorionic gonadotropin, where the mother would ordinarily be producing the hormone, but then at a certain point in pregnancy, the fetus takes over production. The mother produces none. The fetus produces sort of heroic amounts of this hormone. And so we're trying to understand whether the outcomes of parent-offspring conflicts like that might be describable in this kind of framework, right? So if the fetus prefers more of the hormone to be produced than the mother would like to produce, then in this case, we'd expect the fetus, as is observed, to come to produce all of the hormone. So kind of an instantiation of David Haig's loudest voice prevails principle, or here also possibly the cheapest voice prevails. Okay, so just to conclude, given a system with persistent genetic variation, as we would expect if a system is polygenically encoded, system drift is inevitable, our results show. And as I've described, there are many implications for interesting processes in evolution and ecology. Yep, and with that, thank you.